Welcome back to the fifth and final installment in the DNA notes, and this is on restriction enzymes. So restriction enzymes are one tool that are used in genetic engineering, and genetic engineering is a process where we are genetically modifying an organism either to give it beneficial traits, usually from a different species, or to prevent harmful traits. So this could be used, for example, they have taken genes from fish that live in very cold temperatures and put them into strawberries. And so those strawberries are able to withstand freezing temperatures, or at least lower temperatures. Or this could potentially be used in human embryos to prevent genetic disorders such as cystic fibrosis. One tool that you will see often is a plasmid. A plasmid is a small circular DNA molecule found in the cytoplasm of bacteria. So bacteria have one single circular chromosome, then that's where they will carry all of their essential DNA for their life functions. And then many bacteria have one or more plasmids. And on that plasmid, we will have what are called accessory genes, which means that they are not essential for the bacteria to live and function, but they usually carry pretty helpful traits. So for example, antibiotic resistance and the genes that provide certain bacteria with resistance and special proteins and other features to resist antibiotics are usually carried on plasmids. And plasmids can be exchanged between different bacteria, and that is one of the ways that those traits are spread. Plasmids are often used in genetic engineering to transform bacteria, and I will go through this process where we are giving traits of another species to that bacteria cell, usually for laboratory purposes. So our first important step is to cut the DNA. And we do this through restriction enzymes. So they will cut the DNA at a specific nucleotide sequence every time because smaller pieces of DNA are easier to work with, and they're typically named after their source. So for example, EcoR1 is one of the most common restriction enzymes that you will see, and it's named after E. coli bacteria. So you'll see here we have the restriction enzyme. It is cutting at this repeated sequence, CTTAAG, and sometimes you'll see that reversed because it's on both strands. And this is known as a palindrome sequence because it is the same forwards and backwards on each strand. You'll see that it cuts through the backbone on both sides and as well separates the hydrogen bonds between the base pairs. And then we get several fragments on the bottom here, and these overhanging parts are called sticky ends, which we will revisit shortly. So I just thought we would do a sample problem together. You have to exit out so I can highlight for you. So here we have two examples of restriction enzymes. EcoR1, which cuts at this GAATTC, and that would be known as a recognition sequence. And then this is another example, SMA1, and you'll see that this one makes a straight cut. These types of restriction enzymes create what is known as a blunt end, and they're not used as commonly because it's harder to get them to base pair when we are transforming other types of DNA. Or so for example, for making a new plasmid, a blunt end is not as reliable in getting that to work properly. So what we would do is we would look for this sequence here. It could be either on the top or the bottom strand, which of course is just a result of our current orientation. So you'll see we have this in 3 different places. So this enzyme, EcoR1, is going to cut between the G and the A at all 3 of these sites. go back to presentation mode, you'll see here's a answer key I did by hand. So we would cut between the G and the A, and that gives us 1, 2, 3, 4 fragments. So anytime that DNA sequence arises, that recognition sequence arises in the DNA we are working with, that backbone will be cut there. So how do we apply these concepts? So this is an example of how we would make a recombinant organism, in this case a bacterium that contains DNA from a human cell. This is one of the most common applications for recombinant DNA, which is when we combine DNA from different species, and genetic engineering that is used to benefit humans. So we have our bacterial cell on the bottom, and we remove the plasmid from that cell. We're going to use EcoR1 to cut the plasmid so that we can create an opening, and we are going to use EcoR1 on the human gene as well. In this case, we are cutting out the gene for human growth hormone, which certain people sometimes lack and thus prevents them from growing properly. Because we use the same restriction enzyme on both the plasmid and the gene we want to insert, they're going to be able to base pair. So we will mix the plasmid with these DNA fragments, and hopefully some of them are going to combine properly, and there are different techniques for how to sort out bacteria cells that were successfully transformed and see if it worked or not. But once we have isolated those bacterial cells that are now able to make human growth hormone, let me back up. So we mix the DNA and the plasmid, and hopefully some of them are going to combine. We insert that plasmid back into the bacterial cell. We isolate the bacteria that are able to produce that protein, so where our transformation was successful, and then we will allow that bacteria to grow. We will culture it. It will continue to produce that human growth hormone as the bacteria grows and divides normally, and then we will isolate that protein in order to administer to humans who have a deficiency in human growth hormone. And this is also done for insulin. If you know anyone who is diabetic, we used to take insulin from pigs to give to people who are diabetic, but there were a lot of issues of compatibility. Certain people, their body would reject it. But now we're able to make human insulin in bacteria through genetic engineering. So it's a much more pure form, and there's no issue of rejection because it is the human gene and human protein that's being made. So how do restriction enzymes work? Touched on this a little bit, but restriction enzymes cut the phosphodiester bonds, which is the type of bond that we get in the backbone between the sugar and phosphate, to separate fragments of DNA. Ligase is then used to seal the backbone back together. So you can think of it as the glue. Once that plasmid and gene are mixed, find each other, base pair, ligase will be used to seal them permanently. Sticky ends make it easier for the strands to base pair, and any strands cut by the same enzyme will be able to base pair because they have that complementary sequence. So finally, a short video for you all. Hopefully it's not too loud.
Restriction endonucleases are enzymes that cleave DNA at specific nucleotide sequences. The sequence recognized is often 4 to 6 nucleotides long. For example, the restriction endonuclease EcoR1 recognizes the sequence GAATTC. The nucleotides at one end of the recognition sequence are often complementary to those at the other end. The two strands of the DNA duplex have the same nucleotide sequence running in opposite directions for the length of the recognition sequence. Because the same recognition sequence occurs in both strands of the DNA duplex, The restriction endonuclease can bind to and cleave both strands of the DNA molecule. Because the bond cleaved is typically not positioned in the center of the recognition sequence, and the DNA strands are antiparallel, the cut sites are offset from each other. After cleavage, each DNA fragment has a single-stranded end a few nucleotides long, and the single-stranded ends of the two fragments are complementary to each other. These single-stranded ends can pair with each other, sticky ends. Once their ends have paired, the two fragments can be joined together with the enzyme DNA ligase, which reforms the phosphodiester bonds of DNA. What makes restriction endonucleases so valuable is that any two fragments of DNA produced by the same restriction endonuclease can be joined together. Restriction endonucleases are fundamental tools in genetic engineering.
Alright, so go ahead and write your summary and questions, and then I will check them in class.